people, in this video we want to look at the pathogenesis of HIV, that is the human immunodeficiency virus. First, let's take a recap of what we have seen so far. So far, we started off with the human immunodeficiency virus, which can further later lead to AIDS. It later leads to AIDS, okay? So HIV infection is not called AIDS. HIV infection is HIV infection or HIV positive person. Later on, if it progresses, then it becomes a disease called acquired immunodeficiency syndrome, right? Fine. So now let us just continue forward. We saw that HIV virus is an RNA virus. It has an enzyme reverse transcriptase. So it is an RNA virus which has reverse transcriptase. So it goes reverse and becomes DNA in the host cell. The DNA that it makes, that DNA comes and combines with our DNA. And then when our DNA goes through replication, transcription, translation, what happens? The viral proteins get synthesized. Okay, so this is a way of working backwards. So that's why it's called as reverse transcriptase virus or retrovirus. Okay. Now, retrovirus, HIV is not the only virus which is retrovirus. There's another virus called HTLV, that is human T-cell lymphotrophic virus, which is also a retrovirus. The genus is lentivirus. If you want, you can remember that. Okay, this is a small introduction. Then we saw the history of HIV. We saw that the first case was here in New York, and actually how this disease came from chimpanzee, it came to man in rural Africa. The virus then in humans underwent adaptation, right? It went adaptation and it now causes epidemic. Okay, it has now caused epidemic in the world. The epidemiology of HIV we have seen. Basically, the global situation is something like 0.8% adults are affected. That is 35 million people are affected. They are living with HIV. So that is the global situation. And it causes a lot of deaths because there is, as of now, there is no cure to HIV. Now coming to the situation in India, there is 0.27% prevalence. 0.27% prevalence, which is lesser than the global situation. Correct? But in Nagaland, that is a state of India, the prevalence is 0.8, more than that, right? 0.88% is there in Nagaland. So Nagaland has a higher prevalence than the global situation. In India, the states which are affected more are Andhra Pradesh, Maharashtra and Karnataka. Okay? Now, reservoir. Now, who is the reservoir? The infected people themselves are the reservoirs of HIV. High-risk groups are homosexuals, people who have multiple sex partners like prostitutes, etc. Then people who are healthcare workers are also high-risk groups. So I myself am a high-risk group because I'm a healthcare worker. So because of needle prick injury or handling blood products, having an aberration on my own skin, open wound on myself, and then getting exposed to another person who has HIV, all such things are going to make me high risk. Then you have IV drug users, intravenous drug users who are sharing needles and using the same needle again and again. Those people are also high risk group. Then coming to the last here, this one, this is actually telling what, I can't even see it. What is it? Okay, it is about blood transfusion. So whoever is taking blood transfusion, like people who have hemophilia, thalassemia, those people are also high risk groups for HIV. Now, AIDS Control Organization, there are two. In the state we have, in the national level we have NACO, that is National AIDS Control Organization, NACO, NACO, and in the state we have State AIDS Prevention and Control Societies. Prevention and Control Societies, okay, SACS, SACS. In the state we have SACS. Let's move on now to the morphology. Morphology, you have to draw this diagram in the exam, this beautiful circle. So look at the morphology. Let's revise the morphology once. So this is the GP protein, and this is also GP protein, this green and pink. And they form the glycoproteins which are attached to this lipid layer, lipid envelope, okay. Envelope is lipid. Together they constitute the envelope, okay. This GP41 is also called as transmembrane pedicle. Okay, transmembrane pedicle it is called. So this GP120 is actually helping in the invasion, binding to the host CD4 cells and all that. GP20 is the one that will help. Now, and then inside the envelope you have matrix. This thing is the matrix. Then in the center you have the genetic material, 2 identical copies of RNA. 2 identical copies of RNA with the RNA polymerase, right? And around them you have the core, the nucleocapsid, everything you have. So this is the HIV morphology. Now let us move on. Genes and antigens we saw. So there are structural genes which are gag gene, envelope gene and Paul gene. So these 3 are going to generate proteins which form this morphology, which form the structure. Like somebody forms the core, somebody forms the shell. The envelope genes produce these proteins GP120, GP41, right? So these are the genes that produce these antigens or these proteins, okay? In core antigen, you can remember 24. P24 is principal core antigen they are saying. And P18 is nucleocapsid protein. If you want to remember, you can remember this. GP120, 41, this you should not forget. Then coming to the Paul genes which produces the viral enzymes, P51 makes the reverse transcriptase. So can you focus on this one? P51? Say P51 reverse transcriptase, okay? P51 is reverse transcriptase. So we finished the genes and antigens, we finished the structural genes. Now let us move on to non-structural genes. There are a lot of non-structural genes like TAT, NEF, REV, VIF, VPU, VPR, VPX, LTR, okay? What exactly these do are there here. If you want, you can look at it. Otherwise, we have covered it in the previous video also. Just look at it. TAT is transcriptional transactivator gene. It's essential for HIV-1 replication. NEF is negative factor gene. It downregulates CD4 expression. Then REV is regulator of virus gene. It enhances the expression of structural proteins. Then VIF is viral infectivity factor gene. So it influences the infectivity of viral particles. VPU promotes the CD4 degeneration. Okay, CD4 degeneration. TAT, NEF, REV, VIF, VPU. VPR increases the transport of viral genome into the nucleus and also arrests host growth. VPX is found in HIV-2 but not in HIV-1 and in SIV. It is closely related to VPR. LTR means long terminal repeat. It is present on both ends, provide promoter, enhancer and integration signals. At least remember the names TAT, NEF, REV, VIF, VPU, VPR, VPX, LTR. Okay. Guys, in this epidemiology, one more point we wanted to add is this: in sub-Saharan African countries, the prevalence is like 1 in 20. 1 in 20 people will have HIV. So be very careful if you are going there or working there. So now let's move on. We saw the antigen, right? Okay, then we will see the antigenic variation and diversity. So basically reverse transcriptase is error prone. There can be antigenic diversity. The most mutations are in the envelope gene. That makes what? GP120 and GP41, correct? So these are the most, they will change so much, you know. So the envelope looks so different, the antigens look so different. So the HIV virus evades human immune. So when somebody says HIV virus, people get irritated because V is already in virus. That's fine. Don't break your head so much. HIV evades human immune, host immune. Now vaccination against HIV is difficult because of this antigenic variation. Now HIV serotyping, you have 2 serotypes, HIV-1 and HIV-2. Guys, we have 2 serotypes, HIV-1 and HIV-2. Under HIV-1 you have MNOP. MNO very common, M is very important also, okay. Now disinfection and inactivation, we saw that household bleach, ethanol, Lysol, formaldehyde, hydrogen peroxide, heating, extreme pH also can kill or disinfect HIV. We didn't go into much details of this, guys. If you want, you can go into details of this. Now, pathogenesis, modes of transmission. Now we came to the exact topic for this video, pathogenesis. So modes of transmission, then we look at how once it enters as receptor attachment, then replication, right? So pathogenesis, we are looking at these three first of all. After that, we look at more details. Okay, so much more details is there. First, mode of transmission, guys. We're looking at mode of transmission. For mode of transmission, look at this diagram from National AIDS Control Organization. This is the transmission of the virus. So pay attention here, transmission of HIV. So blood transfusion risk is the highest. Highest risk is blood transfusion. Okay. And parent to child is next highest risk. Interesting, right? Parent to child has become the next highest risk. Sexual intercourse actually is just 1%, 0.1 to 1% risk of transmission. Vaginal is higher. Anal intercourse also can spread HIV, and oral intercourse also can spread HIV. Injection drug abuse, that is the IV drug users. Then needle stick exposure, like for health workers like me. And unknown also there are there. Unknown. Somehow they got Papa. Okay, so can you revise this and tell what you saw in transmission of HIV? Blood transfusion main, then parent to child is next, then sexual intercourse, that is where it starts, 0.1 to 1%, vaginal, then anal, then oral, right? Then in IV drug abuse, needle stick exposure, unknown. IV and then needle stick exposure, this is where we are, and we are the lowest risk according to this.